Okay, thank you everybody. I'm glad you came to my talk today. I'm Alex Bricco. Uh, this presentation is on a protein optimization evolving tool, POET, and based off genetic programming. To quickly outline what I'm going to be going over, I'm going to start out talking about background. I'm going to talk about the objectives of the research, look at the way POET has been used to improve an existing function, develop a new function, and then talk about some interesting findings and the convergence thereof. So this project is mostly about MRI, magnetic resonance imaging. It's a method of biomedical imaging. We use strong magnetic fields to generate radio waves from hydrogen atoms from the inside of a subject. It's very widely used, 38 million scans being performed annually across the United States. And in specific with an MRI, this project deals with what is called assessed contrast or chemical exchange saturation transfer. So normally in MRI, we are able to selectively get signal from different hydrogen atoms depending on what those hydrogen atoms are bonded to. So we would stimulate, say, the hydrogen atoms on water, and then the hydrogen atoms from water would produce a signal. In CEST, we can send a different kind of pulse, a pre-saturation pulse, to deactivate hydrogen atoms on something that is not water. The hydrogen atoms are then exchanged with the hydrogen atoms in the water, just over time through normal chemical interactions. And that way, when we send the normal scanning pulse on the water, only the unswapped hydrogen atoms produce a signal. Experimentally, what this looks like is we put a sample inside an MRI, and then we do a series of these pre-saturation pulses at different frequencies to tag these different hydrogen atoms. Then we look at the way in which symmetry is formed from it. So if you can see this beautiful shaped curve, this is what it would look like if we just had water. If we went from starting on the black on this end and going over to the blue. But because we don't, we have this little sag down. And so we basically do subtraction and normalization onto what that gap is. And we did what's called the MTR, or the magnetization transfer ratio, which is our measurement of the contrast produced. The assessed contrast mechanism is useful itself because of its use in reporter genes to our lab. It's very nice for using as reporter genes and tracking different biological processes. MRI is nice for this because you get 3 dimensions, non-invasive imaging amongst different MRI reporter genes. CEST doesn't need metal, which is common to the other mechanisms that people make reporter genes in MRI with. Additionally, the contrast is only visible when you apply a pre-saturation pulse, and these pre-saturation pulses are specific to the individual contrast agents. So you can see only one reporter's effect at a given time, or you can see what the image would look like without contrast at all. Right now, the most heavily used CEST reporter gene is the lysine-rich protein, or LRP. It's been used in a variety of different ways to study different systems, such as oncolytic biotherapy or gene therapy in the heart. And it's good at what it does, but as a scientist and an engineer, we have to ask ourselves, can we do better? So we went to the first big step in protein engineering, which is we used directed evolution. A pretty simple process. We start from an initial peptide, and then we applied mutations to that peptide. Lots of them, lots of different ones. We create a large group of these variants. We scan through them using some method of telling how well they're doing at the function we want them to do. The ones that do better, they go on to the next generation, they get mutated themselves. And so it's like in the figure with the fitness landscape, we start at a low point, but then every time we go forward, we get a little better until we eventually get to the best possible peptide. For this project, though, there are some problems with using directed evolution. We don't really have a naturally evolved starting point to work from, although there are endogenous proteins that produce cest contrast, nothing was designed in nature to generate cest contrasts, as the presence of MRIs has not exerted a significant selective pressure on most of nature. Additionally, the screening rate of us looking at these different mutants and different variants is fairly slow. It would take me to scan 10 peptides about 4 hours. Directed evolution is most commonly used in stuff like in analyzing fluorescent proteins, where you can go through literal millions in a few hours. Additionally, the mechanisms by which cest contrast is understood to work makes it so there's a large number of silent mutations, especially because we use a lot of arginine amino acids, which are most likely to mutate into other arginanes. Additionally, we want a way of scanning the wider search space. So if we started out with in this little part B here, if we start out at a local maxima, directed evolution can't leave a local maxima to get to the global maxima, because each of the small mutations to get it there would be themselves silent mutations or worse, and so we'd think we found the answer. And so we really wanted a way that we could look all around the search space to do so, and slowly converge, learning more about the landscape until we find the global maxima. And that is where POET comes in. POET was designed in an ongoing and active collaboration between the Balançon lab and the Galad lab. It uses genetic programming as a method to predict the function of proteins. We look at a single function that we can quantify well, and we generate the series of models. The models are tested by how well they're able to predict the protein's function. The best models that are most accurate, the least error, they go through mutation of their own sort and recombination with other well-performing models to produce another iteration of models. And then it keeps on doing that until we get better answers. The models themselves are composed of tables with motifs and weights. The motifs are series of amino acids that we would find within the peptides. The weights are a number, positive if it helps the function, negative if it hurts the function. And we take the total fitness of a peptide itself. So in this instance, how much contrast it would generate as the sum of all of the weights for the motifs that the peptide contains. After we run it for a long time, we generate a series of peptides from it using the model. And when we have the best peptides the model can produce, we take those and we continue onward. So there are two big objectives that we had moving forward with this project using POET. The first was how we can use POET to improve an existing function. This meaning contrast at 3.6 ppm. The other thing we wanted to look at is whether we could use it to, instead of improve an existing function, develop a new function, which here is contrast at 5.0 ppm. The workflow was such that we generated an initial data set from prior literature for the 3.6 or the 3.6 research for the 5 of these small 12 amino acid long peptides. And we looked at the contrast they generated. We fed that into POET. The POET developed a model. We then randomly generated large numbers of peptides to look at the large number of peptides, and we assess them with the model. We take the best ones from those randomly generated peptides. We have those ordered, and then we analyze them in the MRI. We feed the data from those back in, and we continue the process. So for our work on proving existing functions, we're looking at 3.6 ppm because this is something where we have many more examples from nature. If you can look at the graph, the dark line, the black one, is cell extract. So it's what the assessed MTR would look like just from a normal cell. When you look at all of the proteins that are just casually moving around through it. And so there's a lot of different motifs that we can pick up on there, and it's been fairly well studied. The initial data sets that we generated was from earlier experiments done by the lab and from prior literature. The first generation, I will admit, was not very good. We had one peptide out of all of them that was actually soluble, and it did not function very well. The later generations have seen substantial improvements in the contrast they've generated, as a larger variety of motifs have been able to be analyzed and learned from. And so now we are producing contrast that is substantially higher than the LRP that is used in research today. Now, for developing a new function, we were looking at 5 ppm. And as you can see from the cell extract versus the peptides that we designed, although everything seems to have a lot of valid contrast generated at 3.6, there's much less at 5 ppm. And as such, a small improvement would yield a relatively large increase in our ability to distinguish from a large body or tell which cells we have the contrast agent in. This is also important because we ourselves do not understand completely the mechanism by which the contrast is generated at that point. And so it has a good example of a problem that maybe POET can understand, even if we as scientists do not yet. The initial training data for these experiments was taken from the 3.6. And so there's less generations done of these than there were of the 3.6, since we had to build up our training data set beforehand. Although the first generation did fairly well, especially compared to the training data, later generations have not performed as well. I'm not entirely sure for the reason why on this, but I believe it's because we primarily have negative responses and very few positive ones to learn from in the training data. But on the subject of the interesting things to find from this, the 3.6 peak, the more studied peak, is very heavily associated with amine groups. And the more amine groups that you have within a peptide, the more positively charged and more basic the peptide becomes. And that's what we see in our training data. We see very little contrast over here at the low PI, and we see very high PI, which is a measure of basically acidity or basicity of the peptide. We see the much more basic peptides generate much more contrast. But when we look at the generations from the POET evolution, we see first that the best peptides are not the most highly charged. They are still positively charged, but they're not as positively charged. And we even see a neutral peptide that is able to produce rather significant contrast compared to the training data. Then if we look at the most recent generation that we've done, the most recent peptides we've generated, the theme over here is still that the most contrast is generated by positively charged peptides. But at the same time, we have this one over here, which is acidic, negatively charged. And it produces more significant contrast than anything we had from our initial training data. Another thing worth looking at from this is that prior work on CES reporter genes, we looked at lysine and we looked at arginine, those two basic amino acid groups that we use to charge the peptides and make them so they produce lots of contrast. And so if you follow the K through the generations, right, we can see that it slowly grows more, but in the more recent generations, which have been doing better, you'll note that there's less likely for there to be lysine, which is K, and far less likely for there to be the gray, which is arginine. And this is interesting because it's not something we would have predict from prior work in the field. The last thing I wanted to look at with these experiments involving POET generating cis peptides is how do we know when we're getting close to final answer? How do we know when we're getting closer to that global maximum? To look at this, I examined the results that we're putting out. If we're converging on the best peptide, then we would be converging on all the peptides would start looking similar as we begin approaching that final peak. And the grand time distance is a way of measuring how different the different peptides are from one another, based not just on the amino acid to amino acid comparison, but also comparing things like size and charge and hydrophobicity. Over time, there has been a slight negative trend in the grand time distance, implying that the peptides are getting slowly but surely more similar to one another. That being said, it hasn't fully fallen into that pattern. As you can see, it shakes up and down. And so there's still probably a good deal more evolution that it can go through. Okay, so overall, POET seems to show promise for the development of these cis reporter genes, and it should likewise, from this, prove to be a useful tool in the overall optimization of proteins for scientific use. Additionally, the genetic programming aspect of this, instead of the rational design of these peptides, has led us to find results and patterns that I never would have expected. It helps us learn more about our field itself, as we see the ways in which novel ideas can generate contrast. I would like to say lots of thanks. First of all, without the Bonsoff lab, none of this would have been possible. And so I'd like to thank Dr. Bonsoff and his grad student, Ilya. We also have our collaborators in the McMahon lab and the Farrar lab. And that would be all. Are there any questions?
Thank you for your talk. If anybody would like to ask a question, please feel free to unmute yourself, or you can ask in the chat.
Sorry, I have a question. Is there any computational validation that, for example, you're generating sequences? Is there any, I know this, I think you're just, based on your talk, you're telling that you are validating the functionality based on experiments. So I'm wondering if there is any computational aspect to know that the generated protein sequence is actually a protein. So it's for example, folding properly, or it's it's just in the realm of the parent actually protein.
Okay, thank you for that question. I do not believe we calculate anything from these other than the predicted contrast. We don't have any real checks on knowing if they will fold properly or anything like that.
Yes, thank you.
Yes.
I have a question.
Yes, go ahead.
If I understand right, you're using a model to compute the fitness of your proposed motifs, and then you're taking the best ones and actually measuring their fitness in an MRI. Did I get that right?
We are using a genetic program to identify motifs from our experimental data that we think might create more contrast. Based off of what motifs we think create more contrast, we create actual peptides, and we experimentally test if those peptides function well.
Right. So my question is, what I'm wondering about is, how good is your model? And do you use the output from your actual machine runs to go back and see how how accurate your model was in predicting the fitness of these motifs?
We have not done significant work looking at going backwards and comparing models to models. The models have not done very well at calculating the exact amount of contrast over time. They have done rather well on telling the order in which they go, but the scale is very different as to what they actually get and what is predicted.
Now, that makes me wonder if you can't use genetic programming to refine your model, the model you used in the first place.
Yeah, that is something we have been considering doing. Just at least looking at the results that we would get if we started from the first models we used and just evolved them forward with the newer data instead of beginning again at every new level.
Cool, thanks.
Thank you.
Yeah, I have to say that, so that James's point is actually a really good one in terms of refining the model, particularly if there are any variables in the model that you're trying to set in order to achieve accuracy or anything like that, because you could actually look at, you can do tests back and forth to figure out which tests to do in the real world based off of what parameters seem to make the biggest changes in the model when you're unsure of things. So this is a technique that I know, for example, Josh Bongard has used for a long time in order to improve virtual models of real world systems.
Yeah, that's really cool.
Of course, there's a risk with that, right? I mean, if you've got your test data and you're measuring the fitness, and then you look at the empirical data and you use that to change the model, you might just end up converging to a model that's very good on the test data you started with. Overfitting.
Sure. I mean, presumably you would have to keep getting new test data,
right,
to make that really work.
Right, or randomizing, choosing random motifs. That was a good idea too that they were doing. Mm-hmm. So anyway, there's a risk, but it would be cool.
So I'm a little bit confused about this method because if we look at the data from the MRI, we always compare it to the data that was predicted, right? It's always, so we kind of do this validation and then we take the data from the MRI and we put it into the algorithm to predict the next generation. So this is kind of using the data already to improve the model. And the whole concept of this algorithm is that if we do enough generations, we're gonna optimize the model. Now, if you would like to do this model mathematically, and we've done it before, you can do it to dipeptide, to single amino acid, to dipeptides. I mean, people have figured out the exchange equations long ago. But now if you want to expand it to 10 amino acids, 12 amino acids, then you start to run into problem of, well, the main problem is my intelligence and then my team intelligence. It's just getting mathematically too complicated to do it. So, but we do fit the measurement, the real time or the real world measurement into the model to improve the model.
Yeah, I think I missed that. Could you describe the model for me? I don't have a good picture in my mind of what kind of model you're using. I was thinking a mathematical model, but what are you actually doing?
Give me a second here. Let's see. Yeah, here. Our models are like these series of tables with the different motifs and the corresponding weights. And we're assuming that the total function would be if we look at the peptides and all of the different motifs expressed within them, and we create a sum of all of the weights for those motifs, that it would give us the total function out.
I got that, but how do you estimate the weights? There's surely a model behind estimating the weights for the motifs.
The weights that we use is one of the evolving portions of POET.
Okay.
So the weights are initially random, and then they are refined over time through the different generations. It's one of the more common things.
That's what Asaf was saying. You are literally
yes.
evolving the model for determining the weights.
Yes.
That's cool.
maybe I can also add something. We use really a diverse population at the beginning. It's really the entire space of possible amino acid peptide sequences. And therefore, we are fishing in a large ocean, so to speak. And the convergence, if it happens, as you can see here, is not there yet. I mean, we are far away from convergence. So we haven't yet hit on the best of these models, for sure.
Mm-hmm. Okay, does anyone have any more questions? Okay. Thank you all for coming to the presentation. I really liked the ideas put forward by a lot of you here. It really made me think about a lot of the things that can be integrated in the future. And yeah, thank you all again. Bye.
Thank you for your talk, Alex. Thanks everyone for the conversation. There are comments in the chat, so make sure you look at those, Alex. And I will see you all at the next seminar in 2 weeks.
Mm-hmm.